I did not know that either.
I'm a
Push in there.
full-on deep sea biologist, and I never even thought about that until this moment. Amazing. You learn something new every day.
All right, we're happy with that. Thank you.
A big pen. Or a big whip. Almost have 4 meters up there. Of course, we're on a hill, so.
Fish.
Go ahead, Amber.
Copy. Your fish.
Looks like an eel, cutthroat eel.
What's the best way to tell a conger from a cutthroat?
Conger from a cutthroat.
You good?
I'm actually
Yes.
not sure about the exact characters. I can tell
behind
looking at
here.
this one that it's a cutthroat eel, but it's kind of the thing where I've just looked at a bunch of them and I don't exactly know what the difference is. Jeff is probably shaking his head in the lounge right now, typing. Cutthroat eels, this is Jeff. Cutthroat eels have a notch in the lower portion of the tail. Congers do not. Not a taxonomic distinction, but a visual one. Okay, I feel slightly better. We just flew over a rock pen.
Bonk. Bonk. Dude.
Wow, the abundance
maybe that's more
of
like
sea stars is really interesting.
Another rock pin, more starfish, another primnoid.
Sacrocalic sponge.
I come up the hill a little from you.
Copy.
Try and stay out in front, but just to the north of you a bit.
Can we look at, where'd it go?
I passed it.
No, yeah, this rock.
Okay, Amber, go ahead.
Couple little corals here.
Tiny ones.
Yeah,
Super
probably
cute.
Paramercids. 2 different ones though,
It's like a little nursery.
and an actual cup coral. All right, thank you.
Yeah, what's going on next to it though? The polyps look really similar,
Yeah,
but
so
it's
it looks like they might be true
crusted.
babies.
huh. So we don't have to worry about this at this exact moment, but are we trying to get a rock sample around the summit?
It wouldn't be a bad idea.
Duster, duster, duster.
I don't know what we have open in the way of, I know we're running low on boxes.
Yeah, in starboard bio box E, there is a cucumber, and I was told that it might try to escape.
Okay,
Oh, here.
that's fine. So
Thank
we
you.
have the 3 smaller ones, but flight risks
Yeah.
in the front box. So we don't have a lot of room for a rock, a big rock at least.
Thanks.
There's
Oh,
a
fish?
fish.
Another synaphobranchid eel, looks like.
I'm pushing a bit there for me.
Right, where's this notch?
So the notch at the
Oh,
very
it's
end.
that.
See how
Okay,
it's,
got it.
Yeah. Oh,
Oh, you touched it.
I touched the screen with the pen and the eel flinched. That was spooky.
The power of the telestrator.
Well, you're the one who can actually touch the sea creatures. As scientists, we don't have that privilege. One thing I would love to know about a landscape like this that I am not sure how we would ever explore, but this
Bridge
volcanic landscape looks very similar to something you would see like on the Big Island of Hawaii, all these fresh lava flows, and they form tubes in caves. And I wonder if there's a network of caves just below the surface here that's been protected from sedimentation, hasn't had time to collapse in on itself, and what's hanging out in those caves.
I would almost guarantee that there are intact lava tubes under here somewhere.
It's very, very tantalizing. There's a whole hidden world that's just in front of us, but have to wait for more technology to be developed to explore.
Talk to Dr. Ballard about that. He has a fascination with underwater caves as well.
Yeah, well,
Want to explore them with smaller ROVs, autonomous vehicles.
yeah, I know there's a few people who have worked on that a little bit, exploring underwater caves, the AUVs, but yeah, definitely an unexplored frontier. underwater caves, underneath ice shelves, places that are even hard to access by deep sea standards.
Some of the things that are getting found too as well is how much complexity there is in flow. And so you've got groundwater, not necessarily in these because they're so basaltic-y, but in some places, the Pacific Northwest is where it's being documented the most, is you get actually groundwater flows moving from one seamount to another. So one seamount might be sucking in water into a subsea aquifer, and others might be discharging it. So it can do all kinds of local effects of flow and oxygenation and stuff like that, depending on how the hydrodynamics inside the crust work between regions.
Yeah, that was a really interesting piece of research from Canada. They found a chain of seamounts where water was going in the porous rocks in one seamount and coming out the other side, and the water was only getting heated up a few degrees just by what's called an adiabatic temperature gradient, and that's just the temperature increases as you go deeper into the Earth's crust because of the pressure of the overlying rocks, not necessarily because of a volcanic hotspot or anything. It's just hotter because it's higher pressure, and that water somehow found its way up out of a different seamount, and they found this massive skate egg nursery where the skates were using the slightly higher ambient water temperature to help incubate their eggs. Zoom fish, please.
Roger. Go ahead, Amber.
Copy.
Been seeing quite a few of these small rat tails. And the genera can be tricky to distinguish. So if we can look at its head as closely as possible, that'd be great. That's perfect.
Our audience is getting some feedback on chewing, asking us if it's good, whatever we're eating. I don't know if you guys hear it. It's like,
I don't know. Is someone's mic on and they're chewing gum?
I'm not
I don't
sure.
think it's me. That's a great zoom. Thank you.
Picking up more sponges over here as we're coming along here. Can we get a little bit lower, please?
Sure.
How tall are the sponges?
From this distance, it's hard to tell. Can we look at this, please?
Roger. Come down for me, Jed.
All right. My altitude's single digits.
Better not come down then.
Yeah, you should head north a little bit there, Dan. You're gonna jump off the end here.
These have been relatively common, so if you want to go, we can find another one. I've seen like 10 in the last 5 minutes, if that's not a good spot. Yeah, move on up that line until we'll find another one.
I was up the hill. I'm not sure what I went wandering.
Yeah, I can see 3 right here.
Okay, Jen, you can come up a bit.
Copy.
Nice and easy.
We're going to start heading kind of northeast now, so.
Roger.
On our way to what looks like slightly steeper side of this seamount here, so we'll see what that yields.
Okay, Amber.
Copy. Are we going for a little sea star?
No, the one below it.
Okay.
That guy there.
Okay.
Can we slurp that?
Sure.
All right, ship is stopped. You're good to go.
Roger.
So this is a cup coral, probably in the family Dendrophilidae. They are notoriously hard to tell apart from video.
Okay,
So
go wide, Amber. I'm
we've
gonna
been
spin
seeing-
around into the hill here.
We've been seeing a lot of these over the last few minutes and at this water depth. So we're going to take this sample, so we'll use it as a voucher to understand what all we're seeing here. Granted, there may be multiple species here, and so, but short of having to take a bunch of samples, we'll kind of use this one as a proxy for all the cup corals we've been seeing. We sometimes are a little biased in the exploration mode of getting the unique rares, the singletons and doubletons that we're seeing, and we don't, and then we go to annotate it and realize that we don't have a good ID on some of the more common things we're seeing.
So I know we're doing some eDNA samples as well. Will the samples that we're taking now contribute to that database?
In the big picture, yes. Potentially not directly with the samples we are taking here. Some of the PICRIC project is biased toward fish in terms of what actual genetic regions they are targeting and the primers they're using. And some research that's ongoing with the Smithsonian and NOAA are showing that some of the more common regions for metabar coding and stuff like that are not as useful in octocorals and corals in general. So largely, yes, the samples we're collecting here will be available to be entered into reference libraries to help future eDNA studies. And and certain taxa do have variations in the same regions that the PICRIC project is looking at. It's kind of a taxa by taxa thing on what the ideal sections of the genomes that get targeted for amplification.
Zoom in there for me.
Okay, zoom in.
Push right in.
Want more?
Yeah.
Okay.
That's good, thanks.
What other colors do these cup corals come in?
This is pretty normal.
Like a pinkish color?
They kind of pink. There's a little bit whiter version sometimes, a little bit darker version, but they're kind of all in this palette of clear and pink.
You can find some yellow ones too.
Yeah, that's true.
Oh, yellow?
Ready
I
reception.
don't know, are those officially cup coral? I mean, they're dendrophilids, you're right, but
Yeah.
I don't. Yeah, okay,
I don't know.
yellow.
I guess based on the pictures in the
Yep.
guide.
Like 75% there.
You'll have to
75.
pop it with the lip, and it should pop right free.
Oh. That's pretty quick.
And there it is.
Smaller than I thought.
Yeah, these are tiny. These are very tiny. A big one, a really big one, is like not quite the size of the palm of your hand.
That'd be neat to see these in the wet lab later.
Can we, before we take off, can we take a look, zoom at that, please?
Sure.
Sample number, please.
That is sample number 26 in Slurp 4.
Gotcha. Thank you.
I think it's a baby Anelopsamia.
All right, that's max zoom there.
All right, thank you.
You're good back there, are you?
We are.
Moving on.
All right, I'm going to get the ship moving again.
Roger.
Bridge nav, 60 meters, 005.
Another black coral.
So Brian, what's your favorite coral?
I like the Chrysogorgias. Definitely of the ones. I don't necessarily have a specific one within the family, but I really like that family as a general rule.
How about you, Harold?
Yeah, I mean the Chrysogorgids are pretty hard to beat, but I'm trying to think of a less popular, a more underrated deep sea coral. There's a lot of choices. I think some of these really big arborescent black corals that just look like a tree on the bottom of the ocean. I think those are some of the coolest ones that I've seen. And there's a few that look like that, but
Is that DBL way
I'll say
wonky
black
or what?
corals to be different.
Oh no, you're up high. That's what's up me.
It's like the larger ones, huh?
Come down a bit, Jade.
To what?
20, so I don't know, somewhere in the green there.
Copy.
$15 to 20 should do it as long as I'm uphill there. You should have more under you than delta, but apparently not. What's going on here? I need to come more to the east. Yeah, the hill's kind of behind us. That's behind you. That's the issue.
Can we take a close look at the rock pen, please?
Sure. Okay, Amber, go ahead.
So this is the recently named genus. Oh,
Oh, hello.
And
Hello.
the scale worm.
scale worm. Trying to steal the show. This is the recently named genus Lithoptilum. Litho for rock. And that's significant because most sea pens we find stuck into the sediment, but these are different because they have this suction cup looking base that allows them to
Come
sit
up
on
a bit,
top of a
Jay.
hard substrate.
Copy.
Yeah, so is that circle thing right there? Or
Yep.
what is that? Is that the...
Yeah, that's the base.
Okay.
So that's its attachment point.
All right, science is happy, thank you. How'd you get the other color skin on the chat? Where's the setting?
I don't know. I just logged in and it was like that.
Interesting.
For those who just logged in and joined us, we are at 1,191 meters, making our way up to an unnamed seamount.
Feels like we're going downhill.
Nice big whip coming up.
Wow.
Can we get a partial zoom?
Yeah. Go ahead, Amber.
All right, that should be good, thanks. Crisis stylid. Squat lobster hanging out there too. I was going through some of the samples from yesterday's dive, specifically looking at the squat lobster we got off one of the black corals, and it was so much tinier than I expected it to be. I mean, I know they're generally small, but I was amazed at how tiny it was. Here's another cup coral, very similar to the one we just collected, but
With these whip type corals,
Stop the ship, Chris.
I wonder how much of the spiraling
Bridge nav, hold
pattern
position.
is intrinsic to their genetics and how much is a response to the flow environment.
I think that depends on each taxa.
Yeah.
I'd give a different answer too, because like the chrysogorgids, which aren't necessarily whips, but they have a very specific repetitive pattern and that's diagnostic taxa. I know less about the bamboos. I would say
So,
just
we're going downhill here.
Okay, we can.
You can see Atlantis looking uphill there.
Okay, yeah, no, that checks out for the next. We were just finishing up this one, so
Right.
I'll change heading.
I don't mind going downhill, just letting you know.
No, it's
I gotta
fine. No,
change
I'm
my
kind of just following contours, but for
strategy a
the
little
small
bit.
stuff I can't really predict, so.
Yeah. You can, you just have to use Atlantis on it.
Yeah,
well, if only we had a multi-beam on Atalanta, we'd really know.
that would be really sweet, wouldn't it?
It would be.
I'd be into that.
We just need to, get an IMU and put an orbit with an IMU on Atalanta.
Well, an INS.
INS, sorry.
Yeah. We ain't got time for IMUs.
I don't think I understand enough of the nuance difference.
How much do you want to understand?
Medium?
Okay, 1 second. Let me put in a ship move.
Okay.
Are you ready for that, Dan? Ship
Yep.
move?
Which way are you going?
We'll be doing 030.
Yeah, that sounds better, thank you.
Yep. Bridge nav. Let's do 40 meters, 030. 030. So, an IMU is an inertial measurement unit. So it really only claims to give you attitude measurements, accelerations, and rate of rotations. An inertial navigation system, INS, I guess my laundry's done. An inertial navigation system has sensors that are sensitive enough such that you can integrate the acceleration. So basically you can just feel all the accelerations over time, add them up, and come up with a reasonable estimate of your position over, depending on the quality of the INS, minutes or hours, or if we're like nuclear subgrade, days or weeks.
Right.
So that's really the difference. And generally, an INS would have a fiber optic gyro. So it's actually using the travel time of light through a fiber optic cable to determine how quickly it's rotating.
So short answer is INS is just more accurate?
That's right, yeah.
Okay.
And so the main reason, an IMU on the surface is fine. The thing that you really need the INS for is that fiber optic gyro, which gives you heading by measuring the rotation of the Earth directly.
Got it.
An IMU normally has
Like a GAMS heading input or something.
well, it has like a fluxgate magnetic compass,
Yeah.
and those are just terrible.
Got it. Because like the IMU for the ship has like a GAMS heading input, right?
Yeah,
GPS,
yeah,
GPS
dual
antenna.
GPS,
Yeah.
yeah. No, and on the surface, that's the way to do it.
Yeah.
Which is why you can get away with, which is why you can easily get away with IMU for, just a MEMS like cell phone, like super nice cell phone type IMU. But subsea, there's problems with it. And you also have issues with magnetic interference and stuff in the bottles,
Right.
as I think the ROV team has learned with Atalanta's compass. And you can calibrate it out and whatever, but it's never going to be great.
Atlanta has an IMU.
Yeah. Yeah, and weren't you having issues with the compass for like years?
Yeah, it has an offset that comes and goes, but
Yeah.
when you set it on the deck, it goes wonky.
Yeah. Well, big steel ship.
Yeah.
Not the place to calibrate compasses. But like it's fine for just general orientation and navigation, but
Yeah.
for
close
survey
enough.
grade mapping, it's not going to cut it.
Thank you. I have always basically used those terms interchangeably.
You can. It's fine.
Okay. Except for when you corrected me.
That's right. A teaching moment, right?
We're backtracking, right? Yes, this is the same whip we just saw?
No.
No? Okay.
Well, oh, did you go back a little bit? Yeah, Dan did go back a little bit.
Okay.
I like this whip.
Megan and Harold, when you're watching this, note to self in the future, annotating, this is the same whip.
Yep. Note to future Harold. Don't count this one twice.
It's a massive. We're just going by it on a bit of a different trajectory here, trying to stay uphill of Atlanta.
Ooh, what's that floating? Yeah, it's gone. Never mind. I'm pretty sure that was a Tudiscarium, which is a really cool little creature.
What sort of creature?
It's a giant radiolarian.
Oh.
Which is?
Normally a microscopic plankton, basically, and there is a deep sea macroscopic version. And
That's cool.
Looked like what that was. But it went by so fast, I'm definitely not going to go on record saying that's what it was. Could have been a number of
I'm
things.
just curious, what's the name again? I'm gonna Google it.
Tudiscarium, I think? And don't ask me to spell it.
Seems like there's a lot of microscopic things in this deep sea.
There is.
Or in the ocean in general.
Yeah, there is kind of just deep sea giantism.
Yeah.
Where you see, yeah, things get bigger. Like just think about like giant ice pods, which we don't have here, but like
Yeah.
further north in the Sea of Japan and like the Gulf of Mexico, you get like pill bugs you see on land that are little and a centimeter long, and they're like 15 centimeters or bigger in the deep sea and quite large.
That's cool. I like the giant single-celled organisms. Over.
Yeah,
Yeah.
and then like xenophyrophores and
Yeah.
stuff like that are crazy.
I will put a small asterisk on that and say
Home
that
the DVL when you
the
get a
overall
chance.
trend of body size in the deep sea is towards smaller things, except when you look at certain functional groups like scavengers.
Thank you.
Your scavengers tend to get bigger, deeper, but other things, maybe just worms living in the sand, other fish, crustaceans,
Yeah, you're right.
not necessarily getting bigger.
It is not a universal constant, certainly.
Bridge nav. Let's do 30 meters, 040.
Yeah, since visiting Palau, I learned about another giant single-celled organism, the
Sea pearls.
sea pearls. Yeah, that's
Yeah,
what they're
those were
called.
cool. Is some kind of algae or something? Was that?
Yeah, it's a
that right?
single-celled algae.
Correct.
Do you know about anything more about sea pearls, Quinlan?
No, I didn't even know those existed here.
Yeah, they were pretty cool.
You know, kind of if you ever snorkel in the iridescent, like marble-looking things on the reef, that's
Oh,
what they
yeah.
are. That's the thing that get me about xenophyrophores is that while they're single-celled, they're multinucleated. So they have many nucleuses, even though they don't have individual cells.
Oh, that's
Right.
interesting.
It's sort of like a slime mold in that way.
More cells per cell.
Exactly.
Still maintaining this relatively high density of the white cookie stars. May have slacked off some on this side, so we're coming around. We took over the watch and we were the southeast-ish side of the summit, and we've kind of covered most of the south side and are now starting up the west side of the summit. Looks like another good-sized bamboo whip coming up here. Now we are seeing some variation in water temperature and oxygen concentration over the last hour. Again, relatively small moves, but definitely real changes.
Bridge nav, 30 meters, 040.
These red nematocarcinus shrimp are once again proving to be quite common.
Yeah, they seem to be just approaching ubiquitous in the deep sea. A couple more cup corals, rockpin. These bamboo whips appear to be kind of the dominant dominant coral here. We seem to have lost. We were on the other side, when we were on the west side, excuse me, the east side, we were seeing more of the smaller kind of primnoids and stuff like that, and as we transitioned over here, we seem to have lost a lot of those and are picking up just these occasional very big bamboo whips with small glass sponges.
Could that be due to the current?
Yep, I mean, it's certainly possible. I haven't seen much of a change in the substrate, so something about the physical or chemical oceanographic conditions here. It's one of the reasons I'm watching the temperature and oxygen concentrations as so closely as I am as we're coming around this feature.
It's interesting to think about the growth patterns in the whip corals, because they don't always just grow straight up. Sometimes they grow up and then take a sharp turn and then start growing more horizontally, possibly optimizing for a certain level up in the benthic boundary layer to maximize their zooplankton catching.
Can we take a zoom on that, please?
Roger. Go ahead, Amber.
Copy.
Let me get a little lower now. I can hold that for a second.
This looks like
A little
a
wider, you said?
No, just hold what you got there.
black coral.
Okay.
Yep. That's probably all we need.
Thank you.
Okay, coming wide.
So with all these mapping that we're doing, do any of these maps, are they available to the public?
Yep. Everything we collect here is available to the public, and will get fed into the large archive efforts looking at global seafloor mapping like Seabed 2030 and JEBCO and things like that. That data will all eventually make it into those big compilations. But everything this ship does is public accessible. All the samples we collect are going on this expedition are going to a long-term repository where anyone can request access to them. And then all the digital data will be available in several different archives depending on the data type in the next 3 to 6 months.
Bridge nav, let's do 30 meters, 045. Affirmative.
And then, of course, all the digital data will also be provided to both PICRIC and the Palauan government as well on hard drives. So we'll make sure to leave copies of everything here in country before we depart.
Oh, that's great. One of our viewers asked about what kind of species is a spaghetti monster.
Bathyphysalia, I think. I don't think they're this far west. I was out on Falcor 2 earlier this year, working in the Salas y Gomez Ridge off the coast of Chile, and we saw a couple on that cruise, and then they saw a couple on the following Nazca Ridge cruise to the north, which got some press. They are really freaky looking creatures. We have a cousin of them here we call a dandelion siphonophore, which is another type of benthic siphonophore, but it's much smaller. I think we see one last leg? We did, didn't we? I think we did.
I think we did.
I didn't.
Yeah, I don't think it wasn't on our watch, but I think Hercules found one at some point last leg.
Oh.
But not a flying spaghetti monster, one of the relatives, the dandelion.
They're really hard to get good imagery of.
Yeah, they are.
Saw a fantastic one, I think it was earlier this year. Just happened to be in a spot where I could finally get a good zoom. It's probably...
Does that have distribution?
Just looking at the Wikipedia article.
challenging to get good image. It's so fragile, the slightest bit of thrust from the ROV washes them away.
Monterey Bay. Not much detail on Wikipedia.
Okay.
The Wikipedia article says, this article is about the species of siphonophore. For the deity, see flying spaghetti monster. That's great.
So with all those like public storage that we have, like how much storage do we have on the ship? Or like how much can we hold?
Hundreds of terabytes.
That's
I don't know.
wild.
I don't know off the top of my head. I am more familiar with the data infrastructure on a ship that does very similar work to Nautilus called the Okeanos, and last time I looked, that ship had 250 terabytes of data storage on board. But that was a few years ago, so I don't know if that's been upgraded since then.
That's amazing.
Quarter of a petabyte.
Nautilus has about 400.
Thanks, Rachel.
400. That's a lot.
I wish you could come.
How much data do we typically generate on an expedition?
For an expedition like this, most of the data that we generate is video. So this is a relatively short cruise for us. We usually do a lot of 30-day cruises. A 30-day cruise can easily generate 50 terabytes or so, or maybe 40 to 50 terabytes of high-resolution videos. But there's many different. A lot of our high-resolution sensors would include systems like Norbit. That Norbit's pretty data-intensive. The Triclops wide-field camera array. We've been shooting photogrammetry, so that can easily be, a terabyte per dive if it's a long dive.
Go ahead, video.
It's a lot of data.
Thanks, Rachel.
Thank you.
Rachel's our data engineer on board.
Similar cup coral.
Rachel, are you still messing with cinema camera?
Yeah, I mean it's running the photogrammetry still timer right now. I've been kind of tweaking the focus from down here.
Roger. The video is garlic again, and out of focus.
All right, gotcha.
All right, we're good with
Photogrammetry
this.
is, I don't know if you're getting anything valuable there, but it's making it miserable to use, to make it miserable to look at.
Do you want me to zoom out?
Yes, please. I can put it in the background if you're still testing.
Let's try and throw it on autofocus for a while and see how that does. I was kind of writing the manual focus, but the issue I found was just that a lot of what we're looking at are rocks, which are kind of round, so it was hard to find something that had a really sharp edge to peek up.
Dan just left for dinner, just so you know, Rachel.
That's the problem with being in the corner of the data lab, is you have no idea what's going on.
I just wanted to let you know. But that was very interesting.
Thanks.
Are you running it through that controller, or are you running it through the computer?
No, I brought the controller down here.
Okay.
Yeah, I have NDI tools on my laptop, because I was using the Mac, it has the accessibility features, so you can zoom in.
Oh, cool.
So
I might have to come check that out. That's really cool.
stop by. I'm in the data/video office.
I'll come down after I'm done with dinner.
Ooh, yeah. Let's zoom on this.
Okay.
Anthomastus.
And, push it in there.
Yep, will do.
This is a mushroom coral. When they're retracted, they look very much like something you'd see in Mario Brothers.
So I always mix up, there's Anthomastus and Pseudo-Anthomastus, and it has to do with these sort of like little pimples in between the tentacles. Megan, do you want to explain that a bit more?
Sure. Yeah, so this is an Anthomastus because it doesn't have those sort of pimply spots in between the main polyps. So you're looking on the surface of like the head of the mushroom. And those are the siphonozoids. So like they have them, but they're smaller. And so when it looks more smooth, it's Anthomastus. When it's a little more bumpy, it's Pseudo-Anthomastus. And then there's another genus, the Heteropolypus, that we see often on like Alaskan and the Pacific Northwest. And those have even larger ones that are quite pronounced.
And what was earlier today we saw a really interesting sort of cluster of
Oh, yeah.
Anthomastus.
So those Anthomastus have the really long bases. Those are called Anthomastus tahinotus. And it was interesting to see it as a large cluster like that because we usually see them as singular individuals.
You can come
but
wide.
these
Can we look at
animals
the yellow
can
corals
reproduce
above?
by budding
Or
off
up
of
higher?
the main adult. So
Yeah.
they're probably all genetically related, that little cluster.
You wanted to zoom here?
Any of these, whatever is easier, one of these yellow ones.
All right.
Yeah, that was really cool to see earlier.
Yeah, those were really pretty. And they were really large, too.
Yeah. I think Dan referred to them as dog toys at one point. Now I can't unsee that.
All right, push in there.
Yep.
Yeah.
Yeah, so this looks like something like Paracis almost. It is a type of
octocoral in the family Acanthogorgiidae. Have we collected anything that looks like this before, back row? Do you guys remember?
I
I haven't collected anything yellow on
I think
my
we've
watches.
definitely collected some yellow corals.
Okay, but yellow where it's like a golden yellow or yellow like light yellow?
There's a lot of different yellow ones.
Daisy yellow or sunglow yellow?
Indeed, yes. This is important distinction.
This one
looks different.
This one does look different. I agree.
What was the name you said, Megan?
It is a, I think it's Paracis. Could be possibly, but it's a little deep for that. But it looks like that.
So we could take a snip of this, I think.
All right.
Okay.
Would you like me to zoom out a bit?
You can come wide.
Yep, sounds good.
Asako has confirmed that we've not seen this one yet, or at least not sampled it.
Oh yeah, she'd know. I don't know how Asako does it, but she's almost like always online. Yeah, on our previous dives, when we've seen the yellow corals,
Or you can push back in.
they've been more of like a darker goldenrod color.
That's good, or do you want a bit more?
That's good there.
Okay, just give me a shout if you need more.
Is this going to be a slurp jar collection, or?
Probably, yeah.
Yeah, the slurp will probably be easier, but it's kind of, depends on how big a piece you end up getting.
It's pretty small.
It is pretty small.
Could we use the slurp gun to actually knock a piece off and slurp it up at the same time?
I think it's going to bend.
Is it?
I think
Okay.
yeah.
Okay.
It's pretty flexible. We usually see these like kind of vibrating.
I think I'm just going to get several pieces of it.
Yeah, sounds good. Oh, the whole thing popped
Yeah,
off.
the whole thing.
I'm going to zoom out for you, Jake.
Yeah, you
I
can
don't
come
know if
wide.
that's going to go through the slurp like that.
Did-
so
Yeah, I don't
maybe
think so.
Did you already get a good zoom in on the polyps before we put it in the basket while they're still
We did not. We
still
can do
open
that.
in the, yeah.
Tell me when.
Yeah, you can push in.
Okay.
They're all closed up. Okay.
Yeah, maybe we put it in the starboard side with that sea star that we collected.
We've got several open small bio boxes on the starboard side.
Okay.
Okay.
Just remember the sea
Sea
cucumber.
cucumber is an escape artist.
Yeah.
Did it try to
Little
escape?
sense of
It tried to escape
Yeah.
out of the net?
Did it really?
Yeah, it got out of the net and it was like floating and Jake had to
No.
like tap it down with the arm.
Oh, wow.
That's a surprise.
Nice, Jake.
It was an excellent save.
Get back in the box.
Get ready for it to go again. I feel like it's going to want to...
It'll feel the vibration of bringing the box out and
It's
start
gonna...
its crunches.
So the 3, any buy a box but the first small one. All 3 are open.
Okay.
What the?
And you might want to put a note down to
Go ahead and bump
process
open the
this
box.
one first to take
Alright, and
photos
then
of
bumping
it
it all the
first
way
when you get it
till
on
it's small.
land, because it might
Alright,
oxidize.
Oh, yeah.
Oh, there goes
ready.
the sea
Oh,
cucumber.
no.
Oh, no.
Oh.
Oh, no.
What have we done?
It's another... Jeez.
Oh,
Yeah.
no.
I think it's lost.
Maybe we'll be able to track it down in a minute.
No, it's going to go
Oh,
through the thruster.
yeah, that was not good.
It looks like
Okay.
a back. Is that a piece of coral leaving, or is that just slime from the... I think a little piece of coral just got out too.
Well, let's finish this.
Yeah, I didn't think we should open that one.
Bump out the box.
All right.
For B.
There it goes.
All right, you can close the box there.
Switch back.
Might we spin around and just see if the sea cucumber might be around?
Yep.
You almost had him though when he was trying to put him back in.
Yeah.
That's, some skills.
We'll let it go.
I think I see it.
You looking in the overhead? Oh, oh wow. Is that it?
That's a sponge.
That's a
It's
sponge.
Missing cucumber.
It'll be bigger than that. Yep.
Yeah, I think our viewers had a scare too. Their messaging, go get it quick. So, hopefully. And if you're just tuning in, we were grabbing a sample of a coral, and we were trying to put it in one of the boxes, and a sea cucumber that was inside one of the boxes managed to escape. So we are trying to locate it, hopefully.
I see a little something floating to your port in Atalanta.
Yeah, that thing down there on the bottom I think is sponge,
Yeah,
Jake.
it's a sponge.
Yeah.
I am twisted now.
Yeah. Different angle.
Down a little bit.
So would that sea cucumber typically be swimming, or would it just try to settle back down on the ground?
It is a type of swimming sea cucumber. It may have decided to take off because of, of course, it's stressed out. So it is possible that it is not on the bottom. They have to keep flapping to stay up, otherwise they do sink very slowly, but they do. Well, I think we should probably continue our way around the seamount, kind of contouring here.
Megan? Megan? Can you check with me before you move the ship?
Oh, okay.
And can you stop the ship?
You don't want to go anywhere?
Well, look where I am.
We're going this way.
We're going along contour to the left.
Yeah.
Right there. All right. I just need to know before you move the ship, please. Just communicate.
Oh, we got some viewers from Japan, Romania, Palau, Philippines, New Zealand, Germany.
Wow.
Welcome. Yeah.
It's great to have everybody online.
Yep. Got some people rooting for the sea cucumber.
Yeah, part of me is too.
Yep. Hope you live a longer life.
So, sorry, you moved on a bearing of what?
050.
Roger.
So we've been looking upslope and sort of crabbing along, spiraling towards the middle.
Roger.
Have you been seeing these, Megan, on the last?
The Tritopleura?
Yeah.
We've been seeing like the single stalk, but not the fan version.
This is a really large one.
Yeah, it's a slightly larger one than what we've seen.
So are those soft? They look kind of hard.
So they have sort of a very dense middle axis, and then
We're
the
going to do a
outside
quick
bits are
video
very
swap.
soft and flexible.
Roger.
Looks like a catcher's mitt.
Yeah.
Elsie, we get a couple good stills. Awesome.
Yeah, that's pretty big.
Yeah. That's great. Thanks, Dan.
We are currently at 1,182 meters, 1,182 meters on the seafloor. Slowly going around the seamount, trying to identify some other species while we slowly make our way to the top.
It looks like a Farreid sponge. Is that Farreid, Megan, or something else?
Conolasma.
Conolasma, okay.
Can I do another move, Dan?
Sure.
Are you sure?
You don't need to be smart about it. You just need to communicate. It's standard up here to talk to each other. Especially when I first sit down, I don't know what's going on.
I just wanted to make sure we kept moving along.
Roger.
We had lots of time.
So is this piece live? Looks kind of dead.
I think the polyps are just retracted.
Oh.
Yeah, it looks like the polyps are just retracted.
That's a very irregular spiral pattern. You could see it on the lower camera, sort of straight down.
Yeah.
It doesn't look like a clean spiral.
And very long.
So we've come around just about 3/4 of the way around the top of this seamount. And within this sort of depth zone, we've seen pretty much the same community. Not a bunch of differences between north, south, east, west, as far as I can tell.
There's some subtle differences, though, I would say. We've kind of lost the cookie stars. They're not as
True.
common. We had several Chrysogorgias on the east side that don't seem to be here on the west side. So I don't know if it's a statistically significant, like there's a difference, but there are some subtle differences.
Yes, closer examination will tell.
Yes, and then we'll have to sit down and actually do the math. Like I don't remember, yeah. I'll have to actually do the annotation and check, but I do feel like there is some subtle differences. This is a really valuable data set to see what all sides look like and how much variation there is. I did a project in the south of Howland and Baker in the Phoenix Islands where we did a side, very similar shaped feature but shallower water. And we did compare all the sides, and generally the vertical zonation was the same in terms of like the communities were, where we saw mainly octocorals, then sponges, then kind of small cup corals and some small Paracis type stuff. And so like in terms of big zones, it was pretty similar in terms of the order in which we saw things, but there was a shift. The upcurrent side, let's see, what would I go with? The upcurrent side, the band transitions were shallower than the downcurrent side, and then certain taxa within those zones favored one current regime more than the other. So like the north and the south, which were the higher current sides, because they were the sides the current was going after, had more similar patterns, and then the east and the west.
Roger.
That's very interesting. I wish we had that level of data for more seamounts.
Yeah, and getting ready to publish that paper, I've only found comparable data from like 3, maybe 4 seamounts that have really investigated the different sides of it. For the most part, we do 1, maybe 2 transects at best and call it good, and say that represents that seamount. And the more we look at seamounts, the more variation within them there is. We have yet to find a single model seamount that explains other seamounts, really. They all seem to be unique, given the low sampling sizes we have right now. You think about even the variation we've seen on this leg, on this cruise so far. Like our first dive and this dive, the seamount depths are pretty different, but the overall shape is similar. But we have still seen pretty different kind of benthic habitats across the 2 different ones. And a lot of that's function of depth. This is a 1,180 meter summit, and the other one was a 670 meter summit. But But even like the better, even in some ways, the better comparison or better contrast were the two seamounts we worked on, or ridge kind of features we worked on south of the main Palau Island group, which were closer in depth and had just dramatically different summits between the two.
Yeah, if only we had the time and money to just do a spiral up every single one, like sort of like we're doing now on the top of this seamount.
What I want to do is and this is, you know, several months worth of work, but I want to take an AUV and truly do a low pass photogrammetry of an entire seamount.
Now you're talking.
That would be pretty cool.
And then instrument it all up and down with current and temperature model readers on all the sides and leave them there for 6 months.
Interesting little worms around the mouth.
Yeah, I agree.
Anemone.
Now, is that what it's trying to eat, or some opportunistic feeding from the worms?
I'm leaning towards the latter.
Yeah.
If it were something it's trying to eat, I would imagine that would be more in the tentacles themselves.
However, how are the worms not getting stung?
Ooh.
Good question.
Like anemone fish, crownfish have lots of special biochemical things to make friendly with anemones.
Are you pausing there?
I got one more.
Say
Yeah.
Great shot.
Wow, you can count the segments on these worms. What are we thinking these are?
I'll give you a segmented worm.
And would they be a parasite to the anemone potentially, or?
Potentially.
Anything is fair game at this point.
Yeah. I've never seen anything like it.
Epic shot, though.
Yeah,
That's a nice shot.
Wow. Yeah, that's really cool.
Nicely lined up, too.
That's really cool. I don't think we're going to learn much more from just looking at it.
Okay, go it.
Actinoscypidae, the Venus flytrap.
Can't take it anymore.
Bridge nav, 30060.
gotten a zoom on one of these sponges yet? These look a bit different.
That'd be a good one if you want one.
Sure.
Talking about the one, oh, that one.
Yeah, that one. This one here looks, in the middle looks dead.
That would not be a good one, but we can give it a go. Go ahead, Amber.
Okay.
It's down in a hole, so we're not going to.
Huh. Yeah, this close-up will be useful. You can see some of the smaller structures on it now. All right, we're happy with that. Thank you.
And for our audience who just joined in, thank you for joining us. And we are making our way around the seamount. As a question, so we are looking at the biology side. So what about the geology side?
Well, unfortunately, we're a little light on geologists on board the ship right now. We've only got one, and their specialty is not volcanic seamounts, so I have to admit, I would love a little bit more geological explanation myself. We are collecting some rocks and things like that that will be of use to geological analysis later, but we're actually very ecologist heavy on this particular expedition.
What we do know is that what we're seeing is mostly basalt flows from volcanism, and it's all sort of crusted over in this botryoidal manganese crust that we see on a lot of surfaces in the deep ocean, with a dusting of sediment.
Did we get a lot of sediment samples?
We have not been collecting sediment samples, no, because they're really frustrating to work with unless you know exactly what analysis you want to do with them. The preservation changes, so it's not as easy as grab a rock, and a lot of different people can use the rock for a lot of different reasons. The sediment samples take a lot more care and feeding, for lack of a better word, a lot more preservation, a lot more work, so we're a little more judicious about collecting the sediment samples unless we know exactly what the reason for collecting them is, so we can preserve them in the appropriate ways for the type of analysis intended. A couple more cup corals.
This is another, I was gonna say flytrap anemone, but not so sure. Can we get a zoom on this anemone?
Sure. Go ahead, Amber.
Okay, zooming in.
Hold that. Okay.
All right, that's good enough.
Going wide.
Thank you.
Bridge now, 30 meters, 065.
I want to know where all the ghost sharks are you saw in your baited camera work and why we haven't seen any yet.
Yeah, I don't know. I would love to see a chimera, but maybe they're just skittish.
What are we looking at here?
This is, I'm not 100% sure what group of glass sponges this is right now. It's the one we just zoomed in on a few minutes ago. And then one of these Venus flytrap, you know, part of the group of Venus flytrap anemones. Or this might actually be not.
That anemone looks like a hormathiid.
Kind of looks
which,
like wings.
I don't know. I think there's a few different anemones that we're calling Venus flytrap.
Yeah, there definitely is.
Cup crawler too, I think.
Just saw it close up. Yeah, and there's quite a few cup corals.
I'll come up the hill there for you, Jade.
Is that a coconut? Maybe?
Yeah, it kind of looks like it, huh?
I don't know if the coconuts are just newer or if there's a different community. We don't see the growing worms, I feel like, the same way. I brought up a coconut once, and it had shockingly little living on it.
Huh.
We had stuff living on it, don't get me wrong. But
So is that a coconut?
Yep.
Yeah. So we've been seeing a lot of these woodfalls around Palau.
Copy.
Both logs from trees and coconuts. And these woodfalls can host extremely diverse communities of animals that use microbes to digest the wood. And there's even chemosynthetic pathways that can arise from woodfall communities. And we can see here there's maybe some urchins, barnacles, limpets.
It's like tiny.
Yeah. So we do have some stuff living on this coconut.
Totally.
Xenophyphore next to it. I think that's about all we're going to see.
Thank
Thank you. I love these perfect terraces of basalt. They look so unnatural, man-made.
So we got an hour of playtime left, right, Chris?
Yep.
And how far is waypoint 6?
150 meters.
Let's make one or two more moves around our depth contour to close it up, and then we'll start heading up.
What's our summit depth?
Bridge now. Let's do 30. 090. Summit depth is 1167.
Okay, so not much different.
So for our viewers, the laser you are seeing is about 10 centimeters.
Another rock pen over there. Can we get a look at this sponge, please?
All right. Pushing a bit for me. Whoa.
Some kind of
Was that you or was that me?
basket sponge.
That must have been you.
Are you talking to video?
Yeah.
No, it kind of just all of a sudden went forward, and then I pulled back out.
Right. Okay, you can go tight there.
Okay.
Yep, cup coral and a euplectellid. We've just seen a couple of these over the last few minutes. I wanted to get a good look at them. All right, we're good, thanks.
Okay, you can go wide, thanks. Can you stop him up for a minute, Chris?
Yeah, we're hanging out. We haven't moved in a minute.
Right.
Fish.
I need to
We look at the fish first, since it might get away.
back up the hill. Go ahead, Amber.
Okay.
Another cusk eel, looks like.
Little mysid shrimp there too. I feel like we've seen more mysid today than we have in a long time. Looks like it's a couple small parasites as well.
So this is the family Ophidiidae.
Is it those little things on that side?
Yep. Some type of parasitic something.
Not quite sure what genus, but this is a good zoom. Possibly a Spectrunculus.
We good?
Yep.
All right.
Thank you. I'm excited about that coral.
Ooh.
Wow.
Wow. What's on it?
So it's got a anemone sort of right at the base of it. And some
Push it just a
ophiuroids.
little for me.
Copy.
That's good to hold that, thanks.
This is giving me Paragorgia.
I would agree that's where my brain's going first, yep. But I have been deceived before by these red pink corals.
Quite a lot of brittle stars in there. Got 1, 2, 3, 4, 5, 6, 7, 8, 9.
Jen, you want to drop the lasers for some beauty shots?
Copy that.
Come down just a few meters for me, Jaden.
Copy.
See the immediate relief there. Stopped pulling on Herc's tail.
If you can, get the ISO lower. I think you can bring the f-stop way down too. There we go.
I can, let me try and get the vehicle a little lower there for you.
Okay. Sure, thanks.
Just on that rock right there. And then we can
So these are sometimes called bubblegum corals because when they retract all their polyps up tight, they just look like someone took a whole bunch of bubblegum and like piled it on top of each other. A very lumpy appearance when they get their polyps all tight.
What makes it so red?
So red is a form of camouflage in the deep sea. Red attenuates the fastest of all the visible wavelengths in the deep sea. Why a coral needs that is an open question. There's a lot of color in the deep sea, and there isn't really a definitive answer of why.
That's neat.
But a lot of the mobile fauna and stuff like that do go up into the mesophotic zone, being red is super beneficial. Once you get fully into the aphotic zone, which we're right on the cusp here between the two, that becomes a harder question to answer.
Alright, gotten quite a few pictures. I'm wondering if maybe it's worth taking a snip of this? You think so?
It's pretty definitively Paragorgia.
Right, it's just there's a lot of species.
There are, you're right. I guess we don't have an impediment on opening the starboard box anymore, do we?
Well, the dinner relief took care of that problem for us.
Yep. How are we doing on sample space?
We have starboard box C and D. E was the one where the cucumber escaped, so I think we are
Oh,
safe
it did
to go.
escape?
Yeah, it did.
Yeah, well, we were having
It did escape.
It escaped. Sure, we can take a snip of this.
I'll come around on the other side so I'm not getting yanked around by the tether there.
Yeah, this is a group that, as far as I'm familiar, there is active genetic research happening to try and figure out the classification of the various different species that are quite hard to tell apart in video, if not impossible.
Do you know who's working on that?
I think Santiago was for a time. Not sure if he still is. There's a lot of work in Alaska too.
Yeah, you're right. Rianne might have been doing some of that.
Does it have like a golden base? Is that what we're looking at?
Yeah, it's kind of yellowy,
Yeah.
golden, yeah.
Yeah, so Paragorgia has a more protein-based skeleton.
Can you put Atalanta over Herc?
I can.
Let's get the bouncy there. Not quite. Could use a little bit more under the keel of Atalanta.
Bridge nav, 20 meters, 310.
You good with all the beauty shots there?
Yep, I'm going to take one or two more just since we found a really cool angle here. But you're good to go.
Roger.
So I would say either right here or right there.
All right. Copy.
I guess what angle is easier for you to come at from here?
Won't know until I get there.
Okay. I mean, basically.
One of those two, Bill.
Okay. Yeah. Any 10 or 15 meter, 10 or 15 meter snip bonus points if it comes with a Brittle Star.
Jaden, can you put your finger on that button right there?
Yep. Do you want the starboard port lights on first?
Nope.
Okay.
This is a very dramatic
Ready?
angle. Secret pictures.
Hey, while you're messing with that and we're sitting here, put it on point focus instead of area focus and see what happens.
Point focus.
Yeah, so
where's that?
It should be that one. And go to probably that one. And now hit auto. Click on the coral. And now hit the AF button. Up top like you focus. Now take the shot.
Somebody read the instructions.
It's the same. It's the same symbology as my big camera.
Oh yeah.
Yeah, that probably helped a bit.
Yeah, that looks better.
Cool. Yeah,
We can go ahead. We're goofing with the camera. We've got lots of good stuff. If you want to go ahead and break out the arm.
It should be good now, Jaden. It's just having some grief there.
Okay.
Push in tight for me there, Albert. Right, a little tighter. Somewhere there, Brett.
If you can give me a little bit more, that would like, yeah, there.
Come on. Didn't want to do.
Perfect.
Great. Thank you.
Please fail. Okay, now you can do the light thing.
It's a great close-up.
Yeah, now you can kind of a little
Yeah.
bit see, since we disturbed it, it's retracted its polyps, and you get a better sense of where the bubblegum name comes from. Just like somebody made a statue out of chewing gum.
You can see on the lower cam, both of the anemones have also closed up. Now they look like, I don't know.
What box is
Catcher's
this going
mitts.
to go in? Hold on, dude.
Okay. Boxes C and D are available.
Those are the aft too small.
Yeah.
Mm-hmm. Yep.
Yep.
Okay. Okay, now you can open. Okay. Close, close, close. Thank you.
All right, to the summit.
Amazing.
Do the
That's
summit.
good.